Good morning everyone. Today I am going to explain glycolytic pathway in brief. So what happened when we intake food material? After consumption of food, all the major biomolecules present in our food, that is proteins, carbohydrates and lipids, get completely oxidized into carbon dioxide and water, during which large amount of energy is produced in the form of ATP molecules. So in the digestion of these carbohydrates, proteins and fats, first they get converted into their building blocks, that is carbohydrates into monosaccharides, proteins into amino acids and fats into fatty acids. Later on, these building blocks get completely oxidized into carbon dioxide and water. So during this process, the monosaccharides, especially glucose, get breakdown into pyruvate during glycolysis process. So glycolysis is the catabolic pathway or oxidative pathway which results in breakdown of glucose into two pyruvate molecules. And coming to the location of the pathway, glycolytic pathway is present in all cells in the cytosolic fraction, and it is otherwise known as EM pathway, that is Embden-Meyerhof pathway. Glycolysis is the main source of energy in the tissues which lack mitochondria, that is the tissues like RBC, corneal cell, etc. they lack mitochondria. So glycolysis is the sole source of energy for these tissues, and the experiments on glycolytic enzymes were conducted on skeletal muscle of rabbit. In case of cancer cells, the glycolysis takes place 10 times higher than normal cells. So estimation of glycolytic enzymes, particularly hexokinase, is an indication of presence of cancer in the cells. All intermediates of glycolysis get phosphorylated. So why all the glycolytic intermediates get phosphorylated? There are three reasons. Number one is to keep glucose molecule intact without entering into the blood, to keep glucose molecule intact inside the cell without entering into the bloodstream, because glucose molecule always tends to enter into the blood. So if phosphorylated, glucose become glucose 6-phosphate and neutral glucose molecule get negatively charged by this phosphorylation reaction. So plasma membrane is impermeable to charged molecules. That's why now glucose molecule cannot cross the plasma membrane to enter into the blood. So it remains intact inside the cell. That is one reason of phosphorylation of glycolytic intermediates. The second reason is energy conservation, and the third reason is to get good access to enzymes. That is, enzymes can easily recognize phosphorylated compounds rather than non-phosphorylated compounds. For these three reasons, the intermediates of glycolysis get phosphorylated. Now let us see the pathway of glycolysis. So glycolysis pathway contains 10 enzymes, out of which 3 are irreversible enzymes and the other 7 are reversible enzymes. For our convenience, we can note the irreversible enzymes by numbers. Enzyme number 1, number 3 and number 10 are irreversible in nature. Other 7 enzymes are reversible. So glycolysis starts with glucose molecules. In the first step, glucose get phosphorylated into glucose 6-phosphate, and this particular step is catalyzed by hexokinase enzyme. Glucokinase can also involve in this particular reaction. These two enzymes differ in their substrate specificity. So the first reaction requires one ATP molecule and magnesium ion. Coming to the second reaction, glucose 6-phosphate get converted into fructose 6-phosphate by phosphohexose isomerase. Here isomerization of glucose 6-phosphate into fructose 6-phosphate takes place, and it is a reversible reaction. And again the third reaction is irreversible reaction, that is conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. Here this reaction is catalyzed by phosphofructokinase, and this is the very important reaction of glycolysis because it is the rate limiting step of glycolysis. Actually all three irreversible steps, that is step number 1, 3 and 10 are regulatory steps in glycolysis, but especially third step is rate limiting step. And also here we can note one point. Here we are calling this compound as fructose 1,6-bisphosphate. Bisphosphate means 2 phosphate groups. Diphosphate also means 2 phosphate groups. But why we are calling here this compound as bisphosphate? Because when we see ADP molecule, adenosine diphosphate, here we are calling it as diphosphate. Diphosphate means 2 phosphate groups are attached to the same carbon side by side. But in bisphosphate, 2 phosphate groups are attached at 2 different carbon atoms. That's why here it is called as fructose 1,6-bisphosphate. At first carbon and 6th carbon, 2 different phosphate groups are attached. So coming to the fourth step, here fructose 1,6-bisphosphate get cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are 3 carbon compounds. So this step is catalyzed by aldolase enzyme, and immediately dihydroxyacetone phosphate get isomerized into glyceraldehyde 3-phosphate by phosphotriose isomerase. So now from glucose molecule, 2 glyceraldehyde 3-phosphate molecules are produced. Now onwards all reactions we have to count by number 2. So 2 molecules of glyceraldehyde 3-phosphate and 2 molecules of 1,3-bisphosphoglycerate, like this up to pyruvate. So now glyceraldehyde 3-phosphate get oxidized into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So in this reaction dehydrogenation takes place, that is the 2 hydrogen molecules are removed and these 2 hydrogen molecules are accepted by NAD to form NADH. Next 1,3-bisphosphoglycerate get converted into 3-phosphoglycerate by phosphoglycerate kinase enzyme, and this is also one of the very important reaction of glycolytic pathway, because here a high energy phosphate group is formed, and from this high energy phosphate group ATP molecule is produced directly from the substrate without involvement of electron transport chain. So This phosphorylation is particularly known as substrate level phosphorylation. So step number 6 of glycolytic pathway is an example for substrate level phosphorylation. In oxidative phosphorylation, from ADP, ATP is formed during electron transport chain, but in substrate level phosphorylation, without involvement of electron transport chain and ADP, directly from the substrate ATP is formed. So in the next, that is eighth reaction, this 3-phosphoglycerate get converted into 2-phosphoglycerate by mutase enzyme. And also this 7th reaction, which is a substrate level phosphorylation, is it has another important feature, that is it is catalyzed by phosphoglycerate kinase, and this reaction is a reversible reaction, an exception of kinase reactions. That is, all the reactions catalyzed by kinases are irreversible in nature, but in this particular reaction it is reversible reaction. So step number 7 is example for substrate level phosphorylation and also exceptional case of kinase, that is it is reversible reaction instead of irreversible. So step number 8 is conversion of 3-phosphoglycerate into 2-phosphoglycerate, which is catalyzed by a mutase, and in step number 9, 2-phosphoglycerate get converted into phosphoenol pyruvate by enolase enzyme, and in the final step, phosphoenol pyruvate get converted into pyruvate by pyruvate kinase. This step is also an example for substrate level phosphorylation, that is another ATP molecule is produced directly from the substrate. So now let us calculate the energetics of glycolytic cycle. In the first and third reactions, 1 ATP molecules are utilized. So 2 ATP molecules are utilized in glycolytic pathway. And coming to the gain of ATP molecules, how many ATP are produced in glycolysis? In step number 6, during the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, 2 NADH molecules are produced. From these 2 NADH molecules, 2 into 3, 6 ATP molecules are produced during electron transport chain. From 1 NADH, 3 ATP molecules are produced in ETC. This is for older version of calculation. And next, in the seventh reaction and 10th reaction, 2 ATP molecules are produced. In total 4 ATP molecules. So in total 4 plus this 6 ATP coming from 2 NADH, 10 ATP molecules are produced during glycolysis pathway. By substituting the 2 ATP molecules which are utilized in step number 1 and 3, the net gain of ATP molecules during glycolysis pathway is 8 ATP molecules. And this is particularly when glycolysis takes place in aerobic conditions only. Glycolysis can take place in aerobic conditions and anaerobic conditions also. When glycolysis takes place in aerobic conditions, the net gain of ATP molecules is 8 ATP molecules. If glycolysis takes place In anaerobic conditions, it results in the formation of lactate instead of pyruvate, and the net gain of ATP molecules is 2 only. Only 2 ATP molecules are produced when glycolysis takes place.